Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to Genetics. In this section, we'll be discussing recombinant DNA and its applications. And as you can see in this particular session, we'll focus on the amplification and manipulation of DNA. So before we get into the meat of this, terms like biotechnology, genetic engineering, they're getting thrown around a lot these days. And it's an exciting time when these things are infiltrating even into the general culture. But I think sometimes these concepts or terms are misunderstood, and we would certainly like to make sure that we start right at the beginning till we get into the more technical details about this. So we need to start by thinking about basic tools and techniques in molecular genetics. Molecular genetics really means dealing with the molecules, perhaps obviously, involved in genetics. Often DNA is the thing that comes to mind. So the term biotechnology, first of all, I say here it's a new revolution, but I just wanted to note that in theory, biotechnology has been going, I should say, has been being practiced by human beings for quite some time. Um, Selective breeding, just as one example, when human beings started to take wolves and breed them in a selective way, the result was dogs, and dogs of all different types, right? So, in a sense, that's using some very rudimentary knowledge about biology to do something. It's applied biology, which is really what biotechnology is. You could say the same about agricultural crops, etc. So, in that very broad sense, biotechnology has been around for a while. Usually, when people say it these days, they mean biotechnology using recombinant DNA. So, this is another very important concept and another important term. Recombinant DNA, we've already heard, a lot about recombination, the general idea of making new combinations of things. This means something, it's the idea is similar, but it's specifically different. So as you see here, this use of the term recombinant DNA, refers to the stable joining together of DNA from different species to make what I refer to here as an unnatural new molecule. The term unnatural seems to have a negative connotation to most people, and I understand why, but I mean it in the most literal and pure sense. Unnatural means something not found in nature, right? So certainly in nature you wouldn't find an E. coli bacterium with a human insulin gene sitting in the middle of it. But that would be an example of a bacterial genome that had a human gene in it, or a mouse genome that had a human gene in it. That molecule would be a recombinant DNA molecule that we have created. And this is not really the focus of this, but it makes some people a little bit nervous that we're creating these things that seem unnatural to them. And I'm not even really making fun. There are some potentially valid concerns about this, but for a good many years now, people have been doing it on a routine basis, and this type of recombinant DNA technology is, considered to be very safe, and maybe we'll see it as natural as time goes by. So there are a lot of things you can do, obviously, and we're going to focus on some of the most fundamental and basic ones. So I say here that many procedures are routinely used with this technology, but of course it depends on what it is you want to achieve, and many tools have been discovered. Discovered is important because we didn't really invent these tools, we discovered them. What we invented is a methodology of using them to achieve our ends. So we have the tools that make the manipulation of genes feasible. Something, let's face it, nobody even really understood what DNA was 50 years ago completely. So, even when they discovered that, I suspect that the routine way that we manipulate genes today would have seemed pretty far into the science fiction sort of world. Um, and I guess that's the next point, is that things are possible today that really didn't seem feasible not that particularly very long ago. All right, so we're going to take a good amount of time and focus on a very basic and important procedure called gene cloning. Now, first of all, before we even get into it, unfortunately, sometimes terminology is used in different ways. So this is really not the same as when you hear about cloning in the sense of, like, many of you may know about Dolly the sheep, a sheep that's often given credit as being the first actual animal that was cloned from another animal. You hear about human cloning. Imagine if I could make another one of me and I could get a lot more done. Those are things that actually are potentially feasible, but it's not really what we mean by gene cloning. Cloning in the most general sense just means sort of copying. So in the case of gene cloning, what it really means is this, and it's very definite about what it means. So when you clone a gene, you're taking some gene of interest. It could, in theory, be anything from a particular organism. So as we go along through this example, I'm going to use the human insulin gene because I like it, and also it's one of the first human genes that was cloned and manipulated and all sorts of good things. So we want to get a hold, as it were, of that human insulin gene, splice it into a cloning vector, which we'll discuss a little bit more in a moment. But what a cloning vector is in its most fundamental sense is a molecule from another organism, often a bacterium, that's capable of replicating in a host organism. And we're going to join those two together to make a recombinant DNA molecule. Right? In this case, it would be one that has the human insulin gene popped into a vector that's composed of genes from another organism. When you take that recombinant DNA molecule and introduce it into a compatible cell, it will then be capable of replicating itself along with that cell, and now you have a way of literally copying the gene of interest. What cloning really does, I mean, you might say that's great, but what's the point of it? There are a lot of, it's a general process. It can be used in the context of a lot of different overall procedures. But really, the way I look at it is cloning allows you to isolate a gene of interest, characterize it. In other words, one thing you might want to do is sequence the DNA there. So it's sort of like, I say, this may sound a little odd, but you've gotten the gene out of the big, it's not really a mess, but when you approach it from our eyes, the whole human genome, it's got, billions and billions of base pairs around it. We just want one little gene. So we took it out, we put it in what I call a safe place. And when it's in this safe place, you can make more of it anytime you want. You can use it for whatever you want. You have cloned the human insulin gene in this case, or whatever other gene you might be interested in. So let us talk about some of the tools that are necessary for doing this. And we need tools, obviously, but in this case, there are a lot of tools, as I said before, that we've discovered. We have to manipulate them properly, but we get them to do a lot of the work for us. And one of the most important tools that was one of the first discovered that allowed this whole revolution to continue, they're called restriction enzymes. Now, officially, they're called restriction endonucleases because what they do is they cut DNA in a very specific way in the middle. That's what an endonuclease means. I mean, as opposed from chopping it from the end. They cut or cleave DNA at very specific sequences, which is great for us because, as you're going to see, we need this ability. You'll sometimes see a restriction enzyme referred to as molecular scissors. I read a newspaper article and it said, scientists use their molecular scissors to cut out a gene from one place and put it into another place. So obviously they're not really a scissors. It's too small, but the idea is sort of similar. So what are restriction enzymes? Where the heck do they come from? They are naturally occurring, so that's why I say we discovered them. They're usually found naturally in bacteria, and right now, hundreds, if not thousands, are commercially available. So you can open up a catalog from whatever your favorite biotechnology store is and say, yeah, I'd like to get that one that cuts at this particular sequence. And that's a very important point. Each different restriction enzyme cuts or cleaves at a very definite and specific DNA sequence. So for whatever sequence it is that you want to cut, We'll see in a minute why you might want to do that. You can usually find a restriction enzyme that will allow you to do this. All right? Now, the names of restriction enzymes often sound strange to people who aren't familiar with them. They're named based on the genus and the species of the organism from which they come. In other words, where do we purify them from? And sometimes a strain of that species. So one of the first restriction enzymes discovered, and a very popular one for use, is called EcoR1. So what that means is the ECO stands for E. coli, the organism that we got it from. And in this case, there's so many strains of E. coli that we have lying around. In this case, it was strain R, and it was the first enzyme discovered in E. coli of this nature, so they call it EcoR number 1. There's also EcoR2, etc. There's, you can go down the list if you discover more than one from an organism, but there are lots of different organisms that these come from, and we'll see different naming conventions. But that's the idea. So whenever you see these weird names, you can relate it to the genus and the species of the organism that it came from. Now, the way restriction enzymes recognize a DNA sequence and ultimately cut it is totally key to the reason that we can use these to do what we want them to do. Restriction enzymes usually recognize a very definite sequence. Technically, the type of sequence that they recognize is one that has twofold rotational symmetry. And this really is the correct way to say it, you guys. Some people will say that they recognize palindromic sequences. You may know what a palindrome is, but this is really important because I remember it taking me a little while to sort this out in my head. A palindromic word is one like race car, in which if you read it the correct way from left to right, or you read it the other way backwards, it says the same thing. It says race car forward, it says race car backwards, right? That's really the meaning of a palindrome. But if you will we'll look here at what EcoR1 really recognizes, and you'll see it's not really a palindrome, because look, here's a sequence 5' GAATTC. Now, here's the reason why, obviously, perhaps that particular sequence is not a palindrome, because if you read it one way, GAATTC, it's not the same as backwards, which is CTTAAC. But since DNA is double-stranded, it takes the idea of a palindrome a little bit further, and that's why I like to use the term twofold rotational symmetry, and most textbooks and professors probably do use that term. Because if you look at the complementary sequence, right, you'll find that what's read in the 5' to 3' direction on the top, GAATTC, if you go down to the bottom and you read the 5' to 3' sequence, you have that same GAATTC. So it's not strictly a palindrome, but what it does this way on the top strand is the same sequence as what it does that way on the bottom strand. That's what it means, twofold rotational symmetry. And if someone does happen to say palindrome in this context, you'll know that this is what they really mean. Okay? Now, another really, and by the way, mess around with sequences. You might say, well, yeah, is that really that uncommon? If you just make a random sequence of double-stranded DNA, you'll find that most often they don't read the same way in one direction as the other. Okay, so if you feel like messing with that just to see if it's true, you can. Very, very importantly as well, most restriction enzymes, after they cut in a staggered way, and what that does is leave so-called sticky ends. Amazingly important concept. So let's look at how they cut. Where I wrote this little mark like that, that's where they cut. I'll put an arrow there as well. But first, let's just look at the top strand, GAATTC, in the 5' to 3' direction. The enzyme recognizes that sequence, and it makes a cut between the G and the A, as my arrow has shown. But on the other strand, it actually does the same thing, but look what happens. So now we're going 5' to 3'. It cuts between the G and the A, right? Right there. It makes sense. I mean, it's twofold rotational symmetry, right? But look what this does. In the next picture, you'll be able to see it a little bit better. But it's not cutting at the same point on both strands. It's cutting over here on the top strand and over there on the bottom strand. That leaves overhangs that are complementary to each other. Thus, they're called sticky ends because if left alone, they tend to go back together. But we can use this to our advantage. Okay, let's see how. So here is a much better picture, obviously, than I can draw to explain how DNA is digested by restriction enzymes. So I put in a little bit more fancy. The artist drew this, not me, obviously. Here is the EcoR1 recognition sequence. So it's really this whole double-stranded area here, right? Just to make sure. He didn't put this in here, but we can put it in. That's the 5' to 3' direction, and that's the 5' to 3' direction on the other strand. So we have GAA-TTC going this way, and we have the same sequence GAA-TTC going, that way on the bottom strand. Now remember where the cuts are going to be made. One right there, one right there. So what this is saying is if you take DNA of this nature that has this site on it, incubate it with the restriction enzyme EcoR1, after it does its cutting, here's what you're going to be left with. So the molecule is cut completely apart, but here's what I referred to before as one of the overhangs, and here's the other overhang. And these arrows that the artist drew are meant to show that these, perhaps obviously, these are complementary sequences, right? So hydrogen bonding wise, they quote want to come back together, but they have now been physically separated from each other. This is an amazingly powerful thing. You might not see why yet, but I'll explain the basics of it. If you take your gene of interest and cut it out in this fashion, and you take your cloning vector and cut it with the same enzyme in this fashion, almost automatically when you incubate them together, the sticky ends will cause association of both the fragments of interest and the cloning vector in an almost automatic way. All right, so we're going to see how that occurs in a little bit more detail. We do need more tools though, and we need the restriction enzymes to cut our molecular scissors. We also need cloning vectors. We mentioned that before, but they are absolutely necessary for amplification of recombinant DNA, which is the actual cloning or copying step. All right? I mean, you need it no matter what you do, but my point is these exist in a way that once you have the gene in a safe place, you can make more of it just by growing cells anytime you want. Now, there are lots of types of cloning vectors. The easiest one to understand, I think, and a popular choice, are bacterial plasmids. A plasmid is an extra chromosomal small ring of DNA, all right? And a proper vector has to have the following three characteristics. They have to have an origin of replication. Remember, we talked about DNA replication before, which will allow them to self-replicate in a host cell. So if you think of, once we make this recombinant DNA molecule, we're going to put it into a cell. Once that cell reproduces, we want the recombinant plasmid to reproduce along with it. So it needs to have its own origin of replication so that it will do so. Plasmids do, in general, reproduce with their host cell, but in an independent fashion. Perhaps obviously, but at this point it may or may not be, they have to have at least one restriction site. Often they have a lot more because we're going to need to sort of open up this vector to allow our gene of interest in. And this is a little bit harder to understand for most people. There has to be a selectable marker. The idea behind a selectable marker is, when you really do this, you can't really see what's going on. You're taking your gene of interest. I'll show you the procedure, but you're taking your gene of interest, incubating it with the cloning vector, and hoping that it gets incorporated in there. But the only way you can know is by subsequently growing cells, and the selectable marker is something that's on the plasmid that will allow the cell, only cells with the plasmid in it, to grow. So if they grow, you'll know that you were successful in getting your plasmid into that cell. Okay. Just for the record, a lot of modern plasmid vectors are engineered, and a lot of companies sell them so that they're particularly useful for cloning. You can, just like you can order restriction enzymes, you can go to your favorite store and pick out a cloning vector that you like. A very common thing that's a feature of many of these, some people call them designer multiple cloning region. Some people call it a polylinker, but what it means is it has restriction enzyme sites for a wide variety of popular, ones that are used a lot, restriction enzymes. So what this reduces the need for is to have a different plasmid every time you want to do something different. You can have one that can be used with a variety of different restriction enzymes. All right, so with this background, let's actually go through the procedure of cloning. It could be anything, but we're going to use the human insulin gene as our example. All right, so number one, using an appropriate restriction enzyme, and we'll discuss what appropriate means in a second, but our goal is that we want to cut out the human insulin gene from the whole mass of the human genome, which contains, as we said, billions of base pairs. So I sort of asked the question, which enzyme would you use? And that's where it comes to what's appropriate. So here's the idea, you guys. Say there's this long stretch of DNA, and the human insulin gene is located right there, from there to there, okay? Insulin, we'll write it down there. Now, the sequence of the human insulin gene is whatever it is, based on the protein that it has to make. But by coincidence, there's going to be restriction enzyme sites in that DNA and in the surrounding DNA. So let's just say we'll make a little fake map here, but it'll give you the idea. Echo R1, there happens to be a site here. There happens to be a site here. And maybe there's even a site over here. Another famous restriction enzyme is called BGL1, but everyone calls it Bagel1. Some of these, you just have to get used to the way it's said. But bagel comes from a species of Bacillus, I forget the species name, begins with GL. And let's say that there's a site right here of Bagel1, so you get the idea. So your idea is you want to cut this out. So take your molecular scissors, cut it out on both ends, so we just grab that fragment out of there. If you use Bagel1, you're going to cut the human insulin gene right in the middle. I mean, not exactly in the middle, but you're going to disrupt it. You definitely don't want to do that. So something like Bagel1 is out. And there are computer programs, or you could do it by hand, by eye, and just say, okay, look, I'm thinking of using Bagel1, and you can go through and say, is there a Bagel1 recognition site in the gene? And if there is, you can forget about that. I'm thinking of using Echo R1. Well, look at that. There's no site in the middle of the gene. That's a good thing. And I've made this a little convenient, but there's so many restriction enzymes that usually this does work out. So look, we have one flanking on one side of the insulin gene. We have a site here flanking it. So if we used Echo R1, we can cut out this whole piece which contains the entire human insulin gene. You may note that it does contain some flanking regions, but it's not a lot, and that's okay. Just want to make sure remember, we're taking this, what might be, I don't know, several thousand base pairs, we're taking it out of the 3 billion to put it in a safe place. So you need to select an enzyme very carefully if you don't want to waste your time and money in doing this. But I hope you can see that in this scenario, if you used Echo R1, you'd be able to pop out the human insulin gene. Yeah, a lot of other stuff might be going on too, but we don't care. All right? Okay, at the same time, but separately in a separate test tube, you're going to take your plasmid vector, which you've also chosen fairly carefully, and it needs to have one recognition site for that enzyme, and you have to use the same enzyme. Now I ask, why do you want one and only one recognition site? So let's explore that just a little bit. A plasmid, unlike a human chromosome, is circular in nature. So if there was, let's make an EcoR1 site right here. If there's one of it in there, think of what happens if you cut a circle one time. What you're really doing is opening up the circle. And that's exactly what you want to do, right? You want to open it up, pop that human insulin gene in there, and then paste it back together, if you want to think of it that way. Let's check this out. If there was another EcoR1 site on this plasmid, we'd cut it twice. It would literally cut this circle into two different fragments. We don't want that. We just want to open it up. So the cloning vector must have one site for the same enzyme that you chose. And you might say, well, isn't this difficult to find? Remember, you get something with the polylinker, it's built for this purpose. So it's built to have one EcoR1 site, one BAGL1 site. So whatever it is that the restriction enzyme is going to work for your purposes, you're going to be able to use this plasmid. It's not hard, really. I mean, I'm not saying you could walk off the street and just be like, yeah, I'm going to clone a gene. But what I'm saying is once you know how to do it, the tools are widely available and it's not terribly difficult. All right? Now, hopefully you're getting the idea. You're cutting them both with the same enzyme. They both have the same sticky ends. So now we're going to take our isolated human insulin gene, stick it in a test tube with our cloning vector. Now, we're going to be careful to add a lot more of our gene of interest than we have of the cloning vector. This is a kinetic thing. Because the cloning vector, you could cut it, but because of those sticky ends, it's going to sort of want to close back up. It won't be a covalent closing, but we don't want it to do that. We want the sticky ends of our fragment that we put in to pair with the sticky ends on the cloning vector. So a way to do that is to put in, say, 10 to 20 times more, and that will increase the likelihood greatly. See, this is not the most efficient process, but you really don't need it to work a lot of times. In theory, once is enough. So you want to give yourself the best chance possible. But that's the idea. So I say, then you allow the sticky ends to pair. And, you know, what you're really doing is just putting it in and letting it sit there. You can't see what's going on, but you can test later to see if the outcome you desired actually occurred. Now, the third tool we need is one we've already heard about, which is DNA ligase. You may remember that from DNA replication, ligase seals covalently pieces of DNA that are already next to each other due to hydrogen bonding. So in this case, you can have the sticky ends go together all you want, but you need to use DNA ligase as the glue, as it were. If the restriction enzymes are the molecular scissors, DNA ligase is the molecular glue that puts this all back together. So I'm hoping you've gotten the idea, but we have our gene of interest. Think of it as a line, right? We have our cloning vector. We're going to open up the vector, put in the gene of interest, and seal the gap so that we now have an unnatural recombinant DNA molecule. It is still a plasmid technically, but it's a plasmid that now contains the human insulin gene along with, in this case, bacterial sequences. So you can see why it's a recombinant DNA, right? Voila. So here's a picture which, if you're not getting it yet, I hope this will explain it a lot better. So we sort of have two different things going on at the same time, but we're keeping them separate in the beginning. We have over here a large human chromosome that happens to contain the human insulin gene, right? I forget where it's located, but remember the gene has a locus. It's going to be in a particular place. We take the bulk of human DNA, cut it with the enzyme that we want. Remember, it's going to be the same enzyme that cuts our DNA and the cloning vector. But let's just do what we did and say it cuts there and it cuts there, right? Now look down here. We now have this piece of DNA that's got the human insulin gene on it, and it's got these sticky ends. TTAA, AATT, right? We took our cloning vector, which in this case is a bacterial plasmid, cut that with the same echo R1, and now this is cut and it has the sticky ends. So these fancy, nice arrows that the artist drew are meant to show you that not only are these sticky ends complementary to, well, it's self-complementary, right? Which shows you why this would be able to close back up, but it's also complementary over here and over here as well. I'm going to mess up the drawing, but the point is that this sequence TTAA is complementary to this AATT, and the same for this. So just without any extra effort on our part, at a perhaps low frequency, but every now and then, the human insulin gene with those same sticky ends will insert itself into the plasmid, and after you put ligase in there, you get this recombinant plasmid. And maybe we could have made the colors a little different, but what's in purple here, which is the same purple here, is the human insulin part of this plasmid, and then you have the whole rest of the plasmid. There you have your recombinant DNA molecule. So you've got, I'm sorry to keep using this terminology, but I think of it that way. We now have that human insulin gene in a safe place, this little plasmid, instead of out in the millions and billions of base pairs of the human genome. Okay, so assuming you understand that we now have that recombinant DNA molecule, I say on its own, the recombinant plasmid, sorry about that, is very interesting, but in order for it to be useful, we have to allow it to propagate itself, right? Copy itself in vivo, which means in a living thing, which means we're going to have to take that plasmid and put it into a bacterium by a process called transformation. We discussed transformation briefly before. Remember, it's the uptake of naked DNA by a bacterium. Now again, this is not a very efficient process, but if you put 1 million molecules and you have loads and loads of bacteria, it happens every now and then. So again, you take the plasmid, you take the bacteria, let them incubate together, and obviously there are some technical steps involved here. You just let them sit though. And the hope is that some of them will be taken up by the bacteria. And this is where the selectable marker comes in, because a very common one, just say it's antibiotic resistance. In this case, ampicillin resistance. So what are we going to do? We're going to take all this mixture of cells that we hope some of them have taken up our plasmid of interest, right? But also on that plasmid of interest is an ampicillin resistance gene. And then when we plate these bacteria out, we're going to do it on a plate that contains ampicillin. So check it out. Isn't it true that the only colonies we see growing on this plate will be ampicillin resistant? Because the other ones are dying. And how did they get to be that way? They got to be that way by taking up our plasmid. So it's a marker in the sense that one doesn't really have to do with the other, ampicillin resistance and the human insulin gene. But if it's growing in the presence of ampicillin, it tells us that this whole plasmid was taken up, and our goal was that the plasmid contained the human insulin gene, and now we can isolate these colonies and know that these bacteria have in them this plasmid, this recombinant plasmid that we just made before. And we picked those colonies, and now we really have them in a safe place. Okay, so now you have what you wanted, which is bacteria. First, you had the recombinant plasmid, and you can keep it isolated, not in vitro, but to put it in the cell is how it's going to be useful, because when you want to, so you could store these cells for a long, long, long time at a cold temperature, and then whenever you want, you can grow some up and amplify your plasmid, which basically means, it may not sound that exciting, but if you're doing research on the human insulin gene, you need a reasonable quantity of that DNA, and you may need it over and over again to do your research. So you come in one day and take a few cells like this, grow them up. Bacteria can go from 1 cell to 1 million in a very, very short time period. So when they do that, you just made 1 copy of your human insulin gene into 1,000,000. And now you can, quote, do things with it. What do you want to do? Well, I don't know. But a lot of things people could do is, if the insulin gene wasn't, or whatever gene you're using, wasn't fully sequenced, you could do a complete DNA sequence on it. You can use this to study its function a variety of ways in vivo, or even take it back out and do in vitro studies with it. From a more technological point of view or applied point of view, you can use it to produce large amounts of human insulin in bacteria. Why would you want to do that? Because type 1 diabetics need insulin injections, as you probably know. In the old days, which weren't that old, they had to get insulin from pigs and horses and cows and things like that. Not only were the animals not so thrilled about it, it was expensive, it was laborious, and even though it worked, pig insulin is close to human insulin, but it's not identical. Now, in a facility that literally looks like a beer brewery or something of that nature, you have giant vats of these genetically engineered bacteria. They're engineered in the sense that we made that recombinant plasmid and put it into them. And now what they do is churn out huge amounts of actual, literal human insulin. Because we asked them to. And I'm being a little facetious, but we asked them to by doing this cloning procedure, transforming the cells. And now, does a bacteria know? It doesn't know anything in the sense that we know it, obviously. So it will express whatever gene is there. We just gave it a human insulin gene to express. Now, there are a lot of technicalities in actually doing this. But if you get that idea, that shows you a lot of aspects of the power of this technology. Because I said cloning is a basic, tool. So here are all different things you could use to do it. Research type things, industrial type things. Just the possibilities are limitless. All right, so cloning and subsequent transformation is one really fundamental process. And I thought we would look at one other one in this session, the polymerase chain reaction, or PCR. Because they are related in a sense. PCR is used to very specifically amplify a particular DNA sequence of interest, but it's done completely in vitro. So this cuts out the whole idea of having to grow up any cells, etc. And in many cases, you can use PCR instead of traditional cloning methods, although it depends again what your goal is, but it's definitely less time intensive. That wouldn't make sense. No living cells are necessary. And check this out. From one molecule, you can make 100 million in about 5 hours. That's pretty amazing. So this is a massive amplification that can be done quickly, easily, even easier once you have all the equipment and tools that you need than cloning. Traditionally, though, it's not really used. It could be, but PCR is used when you need a lot of DNA for a particular purpose relatively fast. Okay, they could use it. You often hear about this in a forensic sense. So if who does this? I don't know. Some crime CSI type of people found a DNA sample, for example, at a crime scene. Often they find a very tiny amount. And in order to do the analysis necessary, they need to amplify it. So PCR is a great way to do that. It's not like a human insulin gene that you want to study the function of. You want to use it in this case for identification purposes. But the bottom line is, any DNA sequence you want to make a lot of, you can do it with PCR. And this just, again, it's a basic sort of a procedure which could be a part of protocols for doing all sorts of different things, from research to industry to DNA fingerprinting, etc. But what's important in this capacity is to understand how it works. Now, in order for this to work, you must know the sequence of at least part of a region you wish to amplify. And notice that I didn't say gene, because it might be a gene, but it also might be another region, particularly for DNA fingerprinting. There are special regions that they look at that are not actually genes. The reason you need to know the sequence is, in this case, you need to design primers that sort of delineate the region. So we want to amplify this region. You don't really have to cut anything out. You make primers that are going to start things off that delineate this region. Okay? So here are the tools that you need to do PCR. You need a template DNA, all right, which could be just genomic DNA that you found somewhere. I'm using this forensic thing just because people are into that, but it doesn't have to be. You need primers that you designed. You need all 4 of the dNTPs. Guys, maybe it didn't dawn on you because I didn't say it yet, but what you're basically doing here is a special type of DNA replication in vitro. So the NTPs, what I mean by that are dNTPs are A, G, C, and T of the DNA variety that can become incorporated into a growing chain. And if it's DNA replication, you need a DNA polymerase to do it. Now, it may not be obvious right now why, but a special DNA polymerase is used, and it's called Taq. And the naming convention here, this is not a restriction enzyme, but it's the way a lot of enzymes are named. Taq comes from a bacterium called Thermus aquaticus, which means likes to live in really, really hot water. It's really what it means. So you take all those, put them into a tube together, and then you put that thing into a thermal cycling machine, which almost everybody just calls a PCR machine. All right? So let's understand how this really works, and I would very much advocate that. I believe we have an animation that shows how this works, but if you go online, you'll find tons of animations to give you the idea of how PCR works. But let me give you the idea first. So the logic of what we want to do, guys, let's go through the steps, and as we go through the steps, I'll explain the logic. Number 1, heat at 90 degrees or more, almost boiling water, 90 degrees C, right? Now I say for X minutes because it's not relevant to us. It's 2 minutes or 3 minutes. But in an actual protocol, it would say heat at 90 degrees for 1 minute. Don't worry about how long. The point of this is to denature the strands. DNA is double-stranded, you know that. It wants to stay together. In order for this to work, we need to separate those strands so that each one of them can act as a template for DNA replication. The easiest way to do that is to heat it all up, okay? So at 90, these strands are going to be separate. Remember, all that other stuff is in the tube, too. Now we're going to cool it down to 40 to 50 for, again, however many minutes. Now this is going to allow primers to anneal. Okay, so this is important. Almost like we saw before, you know how we said with the plasmids the sticky ends want to go back together? Well, if you denature DNA and then cool it down, it's going to want to come back together naturally because of the complementarity and the base pairing rules. So for a similar reason that we discussed before, you're going to put quite an overabundance of the primers into this mixture to make it much more likely that instead of the DNA just going back together, that the primers are going to bind to the now single-stranded DNA because we separated it with the heat. And as everything cools down, not all, but you're going to get a significant amount of the primers binding on automatically, guys, because of the complementarity of the primers. Okay? So I'll have one more picture so you'll get it more. But remember, DNA polymerases can't start things off. They need a primer. Naturally, it's an RNA primer, but a single-strand DNA works just as good. Now, we're going to heat it back up, not quite so hot, to 72, again, for however many minutes. This is where the actual replication is going to occur, the strand elongation. Okay? Now, 72 degrees is the optimal temperature for the enzyme Taq polymerase. This is not normal according to us. Our enzymes like to work at 37. They'd be denatured at 72. So just follow me for a minute and I'll explain why we need to do that. The thermal cycling machine is used because this cycle goes on and on and on, 25 to 30 times. The whole cycle, just on average, it might take 10 minutes. So if you do it 30 times, you got 300 minutes, 6, 12, 18, that's 5 hours, right? That's an average PCR cycle. So You could set up the machine, make sure you program it correctly, or you just wasted your time. You could, if you're neurotic, stand there and watch it, but most people watch it a little bit and then either go home, go out, do another experiment, whatever you want to do. At the end of 5 hours when you come back, if all went well, you now have ridiculous quantities of the DNA that you wanted to amplify. And at that point, you need to isolate it because it's, in with all this mixture. You're going to take it out and make sure you have what you wanted. And what are you going to do with it then? Again, whatever you want to do. Sequence it. You have a lot of the DNA. You could do anything you want with it. Now, the PCR reaction was discovered by someone called Kary Mullis, and he said the idea just came to him. Why couldn't that have been me? I don't know. The idea is an elegant and a brilliant one, but originally there were a couple of technical hurdles, and one of them is having to do with the enzyme. They originally did it. They used regular DNA polymerase, and obviously if you heat that up to 90 degrees, it becomes irreversibly denatured. So what they'd have to do is after each cycle, add new enzyme. The discovery of Taq, it's heat resistant. It's stable at very, very high temperatures. So you don't ever have to add any more, even at 90 degrees. I know it sounds crazy. It does not get denatured. That's because the organism it comes from lives in these very hot temperatures. So the protein has a structure that's resistant to heat denaturation. So as I say here, traditional polymerases, you would have to be adding them all the time because they'd keep getting denatured. So the discovery and usage of Taq really made PCR reasonable. The other thing was, without a machine, you literally had to, in the early days, you would go and say, okay, I have 3 hot water baths here. I'm going to put this in 90 for 1 minute. Okay, let me check that. Okay, now I'm going to put it in the next water bath, 40, 72. You sit there doing it. That wasn't very fun, nor was it efficient. So the thermal cycler was developed by Perkins-Elmer Company, and the PCR machine is now a common piece of equipment in most molecular labs. It does it. It has a heat block in there. You put small tubes in there because the temperature needs to change quickly, and you program it to go up to 90 degrees for 1 minute, go down, etc. So the development of the so-called PCR machine, the thermal cycler, coupled with the discovery and usage of Taq polymerase, is what really makes modern PCR so easy and popular. Now here's a more proper diagram, but I really think if a picture's worth 1,000 words, an animation's got to be worth like 1,000,000. So ultimately you'd like to see an animation. It wouldn't make sense for me to sit here and play you one, I don't think, because we're trying to get the theory. So here, let's check this out, and I think it'll make sense. Here's basic old double-stranded DNA, right? Let's say we want to amplify this region right here, whatever it is for whatever purpose. So step 1, heat up to 90 degrees. We've separated these 2 strands. Each of them is eventually going to act as a template. Now remember, we put a bunch of primers, and the primers are designed, guys, they're complementary to the strands. And remember how DNA replication works. What we want to happen is, once that primer is annealed and the enzyme can get on there, we're going to allow DNA replication to occur in that direction and to occur in that direction. Now, there are some technicalities here. If you really think about it, yes, it'll fall off the end and it'll actually go past the end of the spot where you want to delineate, oppositely on both sides. But what you have to understand is, so at 72, this is the DNA here that's been made in purple and blue over here is the new DNA that was made, all right? But as you keep doing this, you have to envision it. So you repeat the cycle. All the new strands now are getting denatured. So this one, for example, starts right there, including the primer. What's going to happen is in the first few cycles, you'll have some extraneous sequences. But more and more and more cycles that you do, the more you're really zeroing in on that piece that you want to amplify. I know some of you may see that If you don't, that's why you have to check out an animation, okay? But this is the theory. You do it over and over and over again. I mean, how many cycles are enough? It's exponential, so 25 or 30 usually gives enough, even if you were starting with one DNA molecule. All right? PCR, similar to cloning in some ways, different in other ways, very popular if you just want to quickly amplify a particular region for whatever purpose. Okay, well, I hope that was an informative and fun discussion of some of the basic ideas and principles and tools and procedures in modern molecular biology using recombinant DNA technology. In the case of PCR, you're not really creating a recombinant molecule, but you are using primers that you designed and they naturally would not be there. So this is, I guess you're not technically building a recombinant molecule, but you're amplifying it using some of the same types of tools and principles that are used in genetic engineering. Anyway, we're going to continue, if you're watching in order. Next, we'll talk about genetic libraries, and I will see you then.